Thanks very much to the organizers for having a session like this and inviting me. And it's, of course, a great pleasure to be here. And before I talk about the subject that I'm presently working in biology, I want to share with you some things about Spencer when he was half his present age, and about Spencer as an advisor. Of course, working with Spencer was a transformative experience in my life. And the first thing that struck me and many other people, I guess, in TIFR when Spencer joined, was that Spencer had very strong views on many things. Very strong views in physics, okay, and about people, about politics, okay, and in physics, for example, his strong views, of course, ultimately sort of shaped mine and other students' tastes in physics and in science. But there were things that he thought were beautiful, and that range was very large, not just particle physics, but gravity, condensed matter physics, biology, okay? And stuff that he thought was garbage, okay? So the word garbage, I encountered this word garbage as being used for physics for the first time, when Spencer joined the theory group. And, it was quite a shock to me that, papers published in respected journals, even textbook material was, sort of garbage sometimes for Spencer. And later on I learned that garbage didn't mean that it was wrong. It meant that it was uninteresting to Spencer at that time. So, but the range of things that interested him was very large, and that washed off on all of us. Because Spencer had very strong views about many things, he always appeared as somebody very tough and intimidating. And he was intimidating, at the academic level and at the personal level, to people who didn't see eye to eye. And so that was a very strong personality that, everybody had to contend with his presence. And he was, so therefore naturally also tough as an advisor. Any new idea that you would come in would essentially be garbage, for him. And he has always been very tough. Not only then, but even after that, he has been very tough. And it's very difficult to please him. And it's not clear whether I've done anything in my life which doesn't fall in that category. So, now, but in spite of the fact that Spencer is very tough, see, he was very considerate, and he is the one who is responsible for reassuring me that I too can do physics. And, that you don't need to be somebody who is very smart and, obviously brilliant and so on. There is scope for hard work. If you work hard, there's scope for sort of character, for for courage. Things like this matter in a creative life. And the fact that with hard work you can build up intuition, okay? And intuition is built from simple examples. Okay? This is what I learned, as other people, Shanks also emphasized. So this is something that that was very reassuring to me, that I can also perhaps play the game a bit, so on. And Spencer himself was a very intuitive person. He always responded with a gut feeling to things, whether it was in physics. So, I was again very surprised. I used to think, my whole way of, I used to think that physics, science is done, very logically thinking, and, you move from step to step, from one considered and well-meditated step to another one, and so on, and that's how scientists think. And it was surprising to me to see someone at his level groping, groping in the dark, sort of just trying to make leaps, not very clear about why this should be correct, but with a gut level conviction that there is something in this question and that we need to pursue it. That's a way of doing science that I learned from. And this gut level response was there not just in physics, but in everything in life. And I'll narrate one incident in which we were going for a hike in the Western Ghats. So Spenta, Avinash, Shanks, I, other people were also there. And you have to take a local from Churchgate to go to Karjat. And from Karjat, you have to take a bus to get to the hills. And then you start, sort of walking up the Western Ghats. And we were in a bus. And I was in the front of the bus, and the other people, Spenta, Avinash, and everyone were in the back. I was sort of separate from them. And I got into a fight with some boys in the bus. So there was some issue of some seat which I had perhaps offered to someone, and they had taken it, and so on. So there was an altercation, and we were sort of voices were raised, and sort of there was a kind of a scuffle. And in the middle of that, I suddenly hear a voice in the back. Spenta had seen that happening. And Spenta shouted and rushed towards the front of the bus, rolling up his sleeves, okay, taking off his watch. By the time he was in the front of the bus, his watch was taken off, and he was ready to land blows, onto the people. So, this is Spenta, not asking why, not reasoning why, who's in the right, who's in the wrong. This is his gut response, and that's it. Okay, so that is Spenta. And so I will sort of recite a doha, a couplet from Kabir, who is a very famous 15th century poet in the Hindi heartland. And Kabir talks about, he's a revolutionary poet, revolutionary ideas for that time, and even for today, in fact. He talks about the relationship between the guru and the shishya, between the teacher and the student. And I think one of his dohas particularly aptly sort of sums up Spenta's relationship. Not sums up, but sort of at least describes part of the relationship. And so this is, Guru Kumhar Sish Kumbh hai, Gadi Gadi Kadhe Khot. Antar Hath Sahar de, Bahar Mare Chot. So, as to explain a little bit, that Guru, the teacher, he is the potter, and the shishya, the pupil, he is the pot, and the potter is sort of removing sort of one by one the blemishes on the pot. Okay? And the second line says Antar Hath Sahar de. Antar is inside, Hath is hand, Sahar de means support. So he supports the pot from the inside, and Bahar Mare Chot, and on the external part of the pot, he's banging the pot to give it shape. So this is typically Spenta, that who is externally extremely tough, okay, but where it matters, where the real stuff comes, he's there to support you. So that is Spenta. Of course, we have became. Of course, due course, very good friends, and shared many things, lots of things that we did together in TIFR, including working for the Group for Nuclear Disarmament, which was a very important educative effort for all of us working there, and many other things. So of course, it's been a great fortune. We've been very fortunate to have worked with Spenta, and of course, at this juncture, wish him and Lena all the best for the future. So now with this, I turn to some stuff that I've been doing recently. And maybe I should mention that my interest in biology, I think there is something common about biology and string theory. So I think both perhaps deal with ultimate questions. So string theory, the ultimate in terms of the basic fundamental of what, space and time are built of, matter. And biology is the other end, the ultimate in complexity. Okay? And perhaps it's the same reason that that attracts some people to both these subjects. So, but I have been trying to think of some biological phenomena in the simplest biological setting, which is bacterial cells. So I will describe to you some experimental results and then briefly some modeling efforts to understand. So there are experiments for bacteria which have been done decades back, and some very interesting regularities have been observed which are still not understood. So, okay, so the work that I'm going to talk about has been done by students Parth Pratim Pandey and Harshant Singh at Delhi University. And this is a slide that tells you about an experiment in 1962 in which single cells, bacterial cells of E. coli are being studied. And on the x-axis is time in minutes, and on the y-axis, on an arbitrary scale, is the okay. So on the y-axis is the logarithm of the length of the cell. So E. coli grows by elongating in linearly in one dimension, and the length, the log of the length, sort of grows linearly with time, which means that the cell is actually growing exponentially. The size of the cell, or the volume of the cell, length and volume, are growing exponentially with time as the cell is born and then divides. Okay, so the cell is growing from birth to division. So this is in a so-called steady state kind of a culture in which the nutrient environment is constant, and the cells are basically doubling, growing and dividing. So the cell doubles and then divides. And between birth and division, it's growing exponentially. So this is then, there are modern experiments which check this out in great detail, single cell experiments in different bacteria. And not only does the size of the cell grow exponentially, here is cell length versus time. So these are different generations of a cell. So a cell grows and divides, and then you track one of its daughters, that grows and divides, you track one of the daughters, and so on. These are the traces that are being plotted. In red is the size of the cell. In green is the population of an internal protein inside the cell. That also grows exponentially with time. And this, I would like to argue, is surprising actually, because ultimately the size of the cell is a consequence of what's happening inside the cell. There are lots of chemicals, thousands of chemicals inside a bacterial cell. They're interacting with each other, and their dynamics is a complicated nonlinear dynamics. Why should such a complicated nonlinear dynamics result in a simple exponential growth of the volume, or of an individual chemical for that matter, in the steady state, okay? And in fact, not all cells do that. So, this is an example of not a bacterial cell, but a yeast cell, in which the length grows linearly with time for a while until it sort of has a stationary part and then it divides, and so on. So this is not universal, but many bacteria, most bacteria that have been studied show this exponential growth between birth and division. So why is that true? And so here, so this is the kind of models that we've been trying to construct, very simple models to explain such phenomena. Here, xi represent all the chemicals inside the cell, okay? I goes from 1 to n, and you write down some nonlinear dynamical equation which has rate equations coming from chemistry, okay? That's what the population dynamics of chemicals inside the cell as time proceeds. In these models, there is one particular population which has been extracted. We call that Z, okay? And this Z is a special molecule which determines when the cell will divide. So the hypothesis is that this Z molecule also grows with time as the cell grows, and when it reaches a certain threshold, which is ZC, then the cell divides, basically. So all the populations get halved. You're looking at a daughter, all the populations get halved. Z can also be halved. It goes to ZB, Z at birth, which could be ZC by 2, but it could also be something different. Experiments suggest that it could be something different as well, could be 0. This triggering chemical could actually be completely degraded when the cell divides and so on. So that's the kind of model. And you can now study both the growth and the division by choosing some specific, making some specific choices of what these, how many variables you have, how many, what the dynamics is and so on. And you can study the dynamics at a deterministic level, at the level of differential equations, but you can also treat the populations as stochastic variables because, these right-hand sides, these functions fi basically come from reactions, and each reaction is a stochastic thing. It has some probability of occurring, so populations can be treated as integers, and you can study the stochastic dynamics of the cell as well. So let me just show you an example. Here's an example that we look at with 3 of those xi variables, x1, x2, x3. I'm calling them P, T, and R. P stands for precursor. These are the small molecules, like amino acids inside the cell. T stands for transporters. These are enzymes, these are protein molecules, large molecules, which are catalyzing the production of these small molecules, okay? And R, which is the ribosome, which is catalyzing the production of proteins, okay? And itself, inside the cell, okay? So I don't have time to go into detail, but these simple dynamical equations basically capture this dynamics. T, the transporter, is catalyzing the production of P, so it appears in dP by dt. It's allowing external food molecules to come in and be transformed into this amino acid, which is going to be the precursor or the building block of the larger molecules. R is catalyzing the production of T from P. P is being used up, okay, and being converted into T, and P is being used up and being converted into R, also catalyzed by the ribosomes. Okay, so the ribosomes are making themselves as well as other proteins inside this. Okay, and then of course these things can be degraded, and the volume of the cell has been taken to be a linear combination of the various populations inside the cell. Okay. And now, so that's the PTR model, and in the Z sector, the dynamics of Z is also, Z is another protein, just another protein, this dynamics remains the same, but Z is another protein which is also catalyzed by the ribosome R, okay? And so when this Z basically reaches a threshold ZC, the cell will divide, and then all these populations, P, T, and R will be halved, and Z will be reset to 0, let's say, or ZC by 2, depending upon your choice, and so on. And that's your model fully specified once you've specified these parameters. And so what happens is that here is a simulation from different initial conditions. So give some parameter values which you take from the literature, okay? In some sense, identifying the constants of the model with experimental values, and then, sorry. So you see that the 3 populations P, T, and R, green, blue, and orange, they change with time. Also Z changes with time, and doesn't matter what the initial conditions are. 2 examples are shown with 2 different initial conditions. They settle down into a periodic attractor. So this is the deterministic simulation in which they settle down into a periodic attractor. And in this periodic attractor, this is the growth part, this is the division part. So the growth division with, okay, constant ratios. The ratios P by R and T by R, they become constant. Z does not become constant because in this particular ZB was taken to be 0, so it doesn't, okay, but the main chemicals inside the cell, which are the body of the cell, they basically their ratios are becoming constant. And you can see that in a generation, the logarithmic plot versus time, log of the populations versus time, within a generation is straight, which means that they are actually satisfying, they're growing exponentially. So we have a nonlinear model, yet it reaches an attractor in which the internal populations are growing exponentially with time between birth and division. And therefore, since volume is also a linear combination, and also you can see that the slopes, so because the ratios are becoming constant, it means that these 3 slopes are the same. All the 3 chemicals are increasing with the same exponential rate. And therefore, the volume must also increase with the same exponential rate, okay? And that's sort of reproduces the observed qualitative behavior. Okay. This is a deterministic simulation, but supposing you treated those differential equations, okay, so I'll wind up quickly, as stochastic differential equations, so then you see that you reach a stochastic steady state or a deterministic steady state, and you get actually distributions of the birth volume is no longer a fixed number which was there in the deterministic simulation, but it has a distribution. The interdivision time has a distribution, and you can see that the interdivision time is longer here and shorter here, right? And this is because the Z molecule, in order for it to reach a certain threshold, there is a first passage time which is now stochastic because the production of Z is stochastic. And because the first passage time of Z to reach its threshold is stochastic, therefore the cell size also becomes stochastic. And nevertheless, the trajectories of the populations are still approximately exponential. Okay? The volume is also still approximately exponential, even though things are stochastic. And so it's sort of similar to what is seen in the experiments. And I will simply give you the reason why you get exponential growth in this model and in a general class of models that one can consider. The reason is that these functions fi that we chose were homogeneous degree 1 functions of their arguments. So if you rescale all the x's, then the functions also rescale in this fashion. So, for example, you can see that that's the structure of these functions because volume is linear, so therefore each term there is actually homogeneous degree 1. And such systems have exponential solutions. Okay? They are essentially, they are linear in a certain aspect, although they are nonlinear systems. They satisfy this property of linearity in this sense. Okay? And therefore, they have these exponential solutions, and it turns out that a large class of these solutions have exponential attractors, not just merely solutions, but also such solutions are attractors. And so, and it turns out that this is something which is generically going to happen whenever you have mass action kinetics in expanding containers where the volume is a linear function of the population. So this is a generic thing that will happen. So that's why you can understand why this property is a generic property that that has possibly arisen in bacteria. And you have, in addition, the model also reproduces fluctuations in the volume. So you can have, people have measured the distributions, the dots here are from an experiment, and this is the distribution of the added volume in each generation. So different cells are different, and this is the distribution of the interdivision time, and the model has one parameter, which is ZC minus ZB. And in terms of that, you can analytically derive a distribution for the added volume, and also derive a distribution for the division time. And it agrees reasonably well with experiment, and further experiments are needed to see whether the model and its hypotheses are correct or not, but there's sort of preliminary things. So basically, there are other things also that the model reproduces, which I don't have time to go into, the so-called adder property of cell division and bacterial growth laws and so on. But so I don't have time to present that, but the main point here is that there can be some very simple mathematical models, coarse-grained models of cells, which can reproduce some of the observed properties, both of averages as well as fluctuations that are observed in bacterial cells. Thank you.